Welcome to today's lesson on the use of meristem stem cells in cloning plants and their benefits. In this lesson, we will explore how these powerful, undifferentiated cells found in plant tissues can be utilized to replicate plants efficiently. We will also understand the advantages of utilizing this method. In our lesson today, we will delve into the nature of meristem stem cells, discuss how they are used in the process of cloning plants, and understand the various benefits that come from this practice, such as disease resistance and preservation of rare species. So far, you have learned that stem cells are undifferentiated cells, which means that they have the potential to develop into different types of cell. You also understand that meristems are the regions in plants where these stem cells are concentrated and are crucial to achieve plant growth. This foundational understanding allows us to explore how we can harness the potential of meristems for plant cloning. By diving into this concept, we not only advance our biological understanding, but also gain insights into practical applications in agriculture, conservation, and horticulture. This knowledge is integral for addressing global challenges such as food security and biodiversity preservation. Let's get started. To start, let's remind ourselves what meristematic stem cells actually are. Meristematic stem cells are the undifferentiated cells that are found in the growing tips of plants, such as the tips of roots and shoots. These cells have the unique ability to divide and develop into any type of plant cell, making them extremely valuable for the scientific process of cloning. Cloning plants using meristematic stem cells is an effective method for creating genetically identical daughter copies of plants from an original parent plant. Let's now go through how the process works together. First, we select a healthy plant with desirable traits, such as disease resistance or other beneficial characteristics. Next, small samples of tissue, specifically meristematic stem cells, are carefully scraped from the parent plant. These cells are then placed in a special culture medium that contains agar, a gelatinous substance that provides the cells with nutrients and the right conditions to grow. The culture medium is often enriched with hormones to promote cell division and growth. Over time, the meristem cells will develop into small plantlets. These plantlets can then be transferred to soil or other growing media to mature into fully grown plants. There are several significant benefits to cloning plants using meristematic stem cells. One major advantage is that clones can be produced quickly and economically. This means we can generate many plants in a short period, which is particularly beneficial for commercial agriculture and horticulture. Another crucial benefit is the ability to clone rare plant species to avoid extinction. By cloning plants that are endangered, we can ensure their survival and maintain biodiversity. Cloning also allows us to replicate plants with specific advantageous traits, such as disease resistance, for example. Suppose that a particular plant has been genetically engineered or naturally possesses resistance to certain diseases. In that case, we can clone it to produce many identical resilient plants. This method of cloning is not just limited to preserving rare species and enhancing disease resistance. It also has other applications. By cloning plants with desirable traits, farmers can improve crop yields and the quality of produce. Scientists can also study cloned plants to understand particular traits better and execute controlled experiments. Before we finish, let's delve into the history behind the use of stem cells in plant cloning. Plant cloning using stem cells might seem like a modern marvel, but its origins actually trace all the way back to the 1950s. Then, a pioneering experiment by the lab of F.C. Steward at Cornell University demonstrated that a single carrot cell could be coaxed to grow into an entire plant. Let's hear Steward explain their research.
Back in the 1950s, the idea of plant cloning was just beginning to take root. At Cornell University, I conducted an experiment that challenged our understanding of cellular potential. Using a single carrot cell, I grew it into an entire plant. This experiment offered one of the earliest examples of totipotency, the ability of a single cell to give rise to a complete organism. The implications of this discovery were vast. Now, from a single cell, we can cultivate entire fields of crops.
One scientist who collaborated closely with Steward at Cornell and was one of the most prominent figures in plant cloning research was Margaret B. Wilkins. Let's now hear her perspective on their innovative research.
The techniques I developed were crucial in advancing our understanding of plant cell totipotency. Through meticulous lab work, I helped to demonstrate how we could harness plant cell potential in ways that were previously unthinkable. Our research laid the groundwork for what would become known as plant cloning. It was incredibly rewarding to know that my efforts contributed to such a groundbreaking discovery, one that continues to shape the way we utilize plants for the common good today.
In summary, the use of meristematic stem cells in plant cloning is a quick, economical, and highly beneficial technique. The process involves selecting a parent plant, scraping meristem cells, growing them in a nutrient-rich culture medium, and then developing them into new plants. This method not only helps in the conservation of rare species, but also allows the replication of plants with desirable traits such as disease resistance, thereby improving agricultural practices and contributing to various scientific research areas. Understanding this process enables us to appreciate the potential of biotechnology in agriculture and conservation efforts. With plant cloning, we have the tools to ensure food security, preserve biodiversity, and enhance the resilience of crops, all in the face of evolving environmental challenges.